I'm David Eberhard, Senior Medical Director in Oncology at Illumina. I'm a pathologist working to develop tests for cancer patients that can uncover gene mutations and genomic alterations in their tumors and provide critical information to help oncologists choose the best ways to treat and manage these patients. Today we'll discuss what comprehensive genomic profiling is, why it was developed, and the benefits that it offers to patients and clinicians. This talk will introduce these areas: challenges for genomic testing in oncology, the paradigm shift of comprehensive genomic profiling, or CGP, CGP test content, and the value of CGP. When we're done, you will come away with an understanding of how CGP can be useful in providing critical information for the care of cancer patients. Medicine in the 21st century is becoming more and more personalized, so there's a focus on developing medicines that are tailored toward individual aspects of each patient instead of trying to treat everybody the same way. Personalized medicines depend on testing a patient for some aspect of their disease called a biomarker. A biomarker test is performed in a pathology laboratory on a blood or tissue sample from a patient, and the test result can be used to predict whether that patient is likely to benefit from a particular medicine. There are over 100 personalized medicines now approved for use, and 50% of new drugs that are now in development are linked to a biomarker, so that number will continue to grow. In oncology, biomarkers often are related to alterations or mutations of genes that cause cancers to grow and spread. There are over 1,000 ongoing clinical trials for cancer drugs that are linked to gene-based biomarkers, and 50 biomarker-linked therapies are already approved. Again, that number is growing rapidly. The presence of certain gene alterations or mutations in cancers can guide the use of targeted therapies whose mechanism of action is directly related to those alterations. There is growing evidence that a given gene mutation may be present in multiple different tumor types, and we're starting to see some drugs being approved across cancer types. So these have a pan-cancer or tumor-agnostic indication for specific gene mutations or alterations. This slide highlights two recent pan-cancer biomarker approvals, microsatellite instability and NTRK fusion. There's also strong evidence that tumor mutation burden, or TMB, has predictive value for immuno-oncology therapies in many cancer types. We'll discuss this a bit more later. In addition, we expect there will be other mutation-specific therapies that will be approved as pan-cancer over the next years. On the other hand, we also now know that within a specific type of cancer, for example lung cancer, different patients have different gene mutations that cause their tumors to grow and spread. These different mutations are linked to different drug treatments. So for a single tumor type such as lung cancer, there are now multiple gene-directed therapies that have been approved, and this number continues to grow. But while the development of these new drugs is greatly benefiting cancer patients, many times there's limited biopsy tissue available to test for all these different mutations, and this is a growing challenge for oncologists and pathologists. Looking at non-small cell lung cancer, there are many targetable mutations already associated with approved drugs. Biopsies from lung cancer can be very small and have limited amounts of tumor for testing. There are several mutations here that are found only at very low prevalence, so the pathologist must make a choice: about whether to test for the more common mutations first and then go back and test for the others if initial testing is negative, or to test for all mutations, even the rare ones, right up front in every patient. For samples with low tumor content that require macrodissection, or small samples such as needle core biopsies or endobronchial biopsies, there may not be sufficient tumor tissue available to test for all these gene alterations, and this could limit the physician's ability to offer some therapeutic options to her patient. And performing these tests would also use up the entire available tissue sample, so that if additional tissue testing needed to be done, a new tumor rebiopsy procedure would have to be performed. Let's look at some examples of presently available tests for gene alterations and their tissue requirements as described in their package inserts or by the labs who perform the tests. If all of these different biomarker tests were ordered for a lung cancer patient, they would need to submit 29 FFPE slides. If these are small biopsy samples, a lab may run out of tissue before running all the tests, so this may limit the available therapeutic options or create the need for rebiopsy, which is painful and risky for the patient and very expensive. I want to emphasize that the panel of biomarkers shown in this example already misses some of the newest and key emerging ones, such as NTRK and genomic signatures. What if, instead of splitting a sample across multiple separate tests, a lab could perform one single test for all biomarkers at the same time? So in pathology today, we are seeing the need for a change in paradigm. With the growing number of approved and emerging biomarkers, as well as recent pan-cancer biomarker approvals, and the fact that biopsy tissue is typically small and limited, these all reinforce the need for labs to consolidate testing and move toward a single multiplex test that is comprehensive and efficient. It seems we're reaching a tipping point in gene mutation testing for oncology. Here is a quote from a webinar given by Dr. Sandeep Patel in July 2018. His opinion is that as soon as there is one more pan-cancer approval, oncology testing will need to move from individual biomarker testing to a consolidated single multiplex test. The evolution of genomic testing technologies, and in particular next-generation sequencing, or NGS, has progressed to the stage where comprehensive genomic profiling, or CGP, has been developed as an approach that can meet our present and future needs for efficient and economical mutation testing in cancer. CGP, based on NGS, is rapidly being adopted by many labs as their preferred platform for testing. Let's look at what's under the hood of a CGP test. For a CGP assay to be able to cover all biomarkers currently present in clinical practice guidelines, as well as those used in clinical trials, it really needs to cover DNA sequence and gene copy number variants, RNA alterations, including gene fusions and splice variants, and genomic signatures that depend on a combined analysis across many gene loci, such as tumor mutation burden, TMB, and microsatellite instability, MSI. Without all these capabilities, an assay would miss important actionable biomarkers. I'd like to discuss some of these types of genomic alterations in more detail because they represent complex biomarkers that are especially suited for testing by next-generation sequencing technologies. First are genomic signatures, such as MSI and TMB, which have become important because of their abilities to predict patients that are more likely to benefit from immune checkpoint inhibitors. Pembrolizumab already has a pan-cancer or tumor type agnostic approval for use in MSI-high cancers. TMB is emerging as another potential pan-cancer biomarker. It's measured by assessing the total number of mutations across a large expanse of coding genome. Cancer cells with a high mutation burden, TMB, are believed to express more neoantigens, which are recognized by immune cells, giving a better response to IO therapies. There are presently over 1,000 clinical trials exploring immunotherapies in various settings, and these biomarkers are being intensively explored in clinical studies. For example, researchers at Memorial Sloan Kettering recently published an important study looking at the relationship of TMB to immunotherapy response in a cohort of over 1,600 patients. This was a retrospective study of patients with several different solid tumor types who received a variety of immune-targeted drugs alone or in combinations. This study found that within each tumor type, the 20% of patients with the highest TMB scores responded better to immune therapies. However, the actual TMB value that separated the upper quintile with greatest survival benefit from those with lower scores and less benefit was different for different tumor types. Thus, this study provides evidence that TMB high can be a general predictor of immunotherapy benefit in solid tumors, but at the same time that the cutoff for distinguishing TMB high from TMB low will likely vary by cancer histology type. Although TMB and MSI are both biomarkers that predict benefit to immunotherapies, they are not interchangeable with each other, nor with PD-L1. We cannot use the status of one biomarker to infer the status of the other. This study, including over 11,000 cases, shows that the most commonly used IO biomarkers, TMB, MSI, and PD-L1, are not well correlated. So a CGP assay ideally should include both TMB and MSI on top of other biomarkers. It is essential to include robust detection of gene fusions as part of an assay. Gene fusions are present in 15% of metastatic cancers, and many of these represent actionable alterations that are targeted by drugs that are already approved and in clinical development. Many methods for detecting gene fusions cannot identify specific fusion pairs or are limited to detecting only certain predefined fusion partners. However, fusions of target genes with novel partners are also therapeutically actionable, and tests that cannot identify these novel fusion partners may be unable to provide important information for making treatment decisions. We want to highlight here the fact that fusion detection is quite complex because typically fusion genes have multiple fusion partners. An important example are fusions involving the NTRK gene family. The NTRK genes are known to have more than 60 fusion partners, and this number keeps growing as new partners are discovered. RET, ROS1, and ALK are other examples of clinically important fusion genes that have many different fusion partners. Detecting both known and unknown fusion partners for these genes is important because they all can be actionable. Analyzing RNA in addition to DNA allows us to detect the broadest number of fusion genes that are actually translated and expressed, and using an NGS hybrid capture assay allows detection of these gene fusions regardless of the fusion partner. We're starting to see that different fusion partners affect the sensitivity of the target gene to drugs, so it will become important to be able to identify the specific partner involved in the fusion to make the optimal drug choice. This table summarizes The abilities of various assay technologies to detect the different types of molecular alterations that are needed for a comprehensive assessment of actionable biomarkers. Genomic profiling using hybrid capture-based library preparation, followed by next-generation sequencing by synthesis, is the only approach that can look into all the gene biomarkers. Of course, protein expression cannot be directly assessed by any tests that analyze DNA or RNA, and likewise, immunohistochemistry cannot directly assess copy number or gene fusions. Comprehensive genomic profiling provides a lot of information. One concern that's been raised by labs and physicians who receive CGP test results is that the reports can be overwhelming and difficult to interpret. This example is meant to show that this challenge can be addressed by thoughtful organization and formatting of the report. It highlights the actionable mutations that can be linked to available therapies and clinical trials, and provides supporting clinical evidence and clear, concise annotation, helping the physician make an informed decision on patient management. Most available reporting software are backed up by a clinical database that contains a lot of curated data to help provide background on the findings from the report. This slide highlights the potential diagnostic yield of a CGP test. These are 4 examples of independent studies that asked what percentage of patients in a clinically defined cohort would be identified with an actionable alteration by CGP testing. The cohorts in these studies were different, and the exact definition of actionable findings were also somewhat different, but these can be generally described as alterations that lead to insights on therapy or possible available clinical trials. Here, even the study with the lowest number shows that nearly 1/3 of patients were found to have actionable alterations, and this ranged up to 90% in this last study. The value of CGP is shown in this microcosting study published by the Association for Molecular Pathology in 2016 that looked into the utility and health economic benefits of incorporating comprehensive NGS testing in patient management. This study concluded that testing patients with NGS would lead to significantly more eligibility to clinical trials and more utilization of targeted therapies, which would ultimately help reduce adverse events and costs associated with non-targeted therapies. The various aspects of CGP that we've discussed today show the potential for major benefits to multiple stakeholders in the medical industry in different ways. CGP enables patients and their doctors to find the best treatments, provides efficiency of time and cost to the pathologist, promotes enrollment into clinical trials for new drug development, and allows healthcare institutions to provide precision medicine to their communities. These benefits highlight the potential for CGP to be a truly transformative advance in the care of cancer patients. These benefits of CGP are supported by considerable evidence that's been generated over the recent past years, and payers are starting to embrace the clinical utility and economic benefit of CGP tests. Now there's a CPT code for CGP and expanded panels, and it's covered by Medicare, Medicaid, as well as many commercial U.S. payers. That means that 160 million out of 309 million insured lives in the U.S. can already receive coverage for a CGP test in the case of advanced cancer. This will be a significant driver for CGP to become standard of care for advanced cancer patients in the near future. Let's review the main ideas that we've discussed today about comprehensive genomic profiling. CGP provides a significant increase in our biomarker detection capabilities, maximizing the chances of identifying relevant alterations the first time. CGP enables consolidation, providing a series of benefits as compared to reflex testing and leading to potential improvements in time to result. The ideal content for a CGP test should include all key biomarkers present in guidelines and clinical trials. A CGP test should use a high-quality and reliable assay capable of gene mutation and signature detection across all variant classes. And finally, CGP has value in healthcare. I hope that this discussion has been valuable in helping you understand the basics of comprehensive genomic profiling and its benefits in the world of oncology. Thank you for your participation.